Law of segregation, and beyond. Remember Mendel's fourth hypothesis? The law of segregation. A sperm or egg carries only one allele for each trait because allele pairs separate from each other during gamete formation. Mendel did not stop there. He devised a second law of inheritance by observing two traits at the same time. For example, seed color and seed shape. Mendel wanted to see if these different traits affected each other or not. In other words, did they behave independently from each other or not? Dihybrid cross. Mendel performed dihybrid crosses, crossing true breeding parents differing in two characteristics, to determine whether the characteristics are transmitted to the offspring together or independently. For example, a pea plant with yellow pods and yellow seeds, little g, big Y, crossed with a pea plant with green pods and green seeds, big G, little y. Such a cross produces an F1 generation which is heterozygous for both characteristics, big G, big Y. We could see this because in each parent, they can only give up one allele for each of the traits. So the parent that has little g, big Y, can only produce gametes that are little g, big Y. And then the other parent that is big G, little y, can only produce gametes that are big G, little y. When we cross these two gametes together, we get big G, big Y, little y. It's important that we keep the same types of letters together. Keep the capital G with the lowercase g and the capital Y with the lowercase y. Dihybrid cross. Crossing the F1 generation produced a 9:3 to 3:1 ratio of phenotypes and a genotypic ratio of 1:2 to 1:4 to 1:2 to 2:1. These gametes we could generate from the F1 individuals using the FOIL method. That's first, outer, inner, last. So if we take the genotype of one of these heterozygous individuals that both have the same genotype, big G, big Y, little y, do first, so it's big G, big Y is one gamete. The outer, big G, little y. The inner, little g, big Y. And the last will be little g, little y. We put those four gametes on each side of the Punnet square, one set of gametes for one parent, one set of gametes for the other, and then like doing the monohybrid cross, we just match up the alleles in each of their individual boxes, remembering to keep the letters together. So keep the G's together and the Y's together. The law of independent assortment. Using a dihybrid cross, Mendel developed his second law of inheritance. The law of independent assortment states that each pair of alleles segregates independently from other pairs of alleles during gamete formation. So in the last example, we saw that we could get different combinations of pea pods and pea colors. This law of independent assortment applies only to genes on different, non-homologous chromosomes. Genes located near each other on the same chromosome tend to be inherited together. The law of independent assortment. The alleles of one gene segregate into gametes independently of another gene's alleles. Think of this like a coin toss. If you toss a coin, what is the probability of it coming up on heads? Now if you toss a second coin, what is the probability of it coming up on heads as well? When tossing a coin, the outcome of one toss has no impact on the outcome of the next toss. Rules of probability. Mendel's laws of segregation and independent assortment reflect the rules of probability. Remember from math, the probability scale ranges from 0 to 1. An event that is certain to occur has a probability of 1. An event that is certain not to occur has a probability of 0. And the probabilities of all possible outcomes for an event must add up to 1. Using probability rules when solving genetics problems. There are two rules of probability that are helpful when solving problems in genetics: the multiplication rule and the addition rule. The multiplication rule. The multiplication rule states that the probability that two or more events will occur together is the product of their individual probabilities. For example, you would use the multiplication rule if you want to know the probability that a plant will have both yellow seeds and yellow pods. So for pea color, yellow seeds, it is a 75% chance that they will be yellow. And for pod color, it is a 25% chance that those will be yellow. So we take 3 out of 4, multiply that by 1 out of 4, and we get a total probability of 3 out of 16. And we can see that corresponds to the Punnet square here, the actual chart, for yellow seeds and yellow pods, that is 3 out of 16. While it is useful to have this Punnet square as a visual reference, if you want to save time to try and figure out what percentage will have yellow seeds and yellow pods, the multiplication rule is obviously a lot faster. The rule of addition. The rule of addition states the probability that an event can occur in 2 or more alternative ways is the sum of the separate probabilities of the different ways. For example, you would use the addition rule if you want to know the probability of a plant produced from a monohybrid cross being homozygous dominant for a flower color or homozygous recessive for flower color. So in this case, it's a 25% chance that the offspring will be big P, and it's a 25% chance that the offspring will be little p. If we want to know the probability of the offspring being homozygous dominant or homozygous recessive, we add them. In this case, it's a 50% chance that the offspring will be homozygous, big P, or little p.